Hello, everyone. Welcome to our Cell Therapy Symposium, which is hosted in collaboration between Proteintech and RoslinCT. We have a really excellent lineup of speakers today who will be sharing their work in the cell therapy space and discussing kind of the latest innovations and their work for process development. Before we get started with the symposium, though, and the speaker introductions, I do just want to share a few slides introducing our company, Proteintech. Oops. So, Proteintech is a leading provider of life science reagents. The company was founded in 2001, and at that time, it primarily was a supplier of antibodies and immunoassays. And so, because we've been in the game for so long, we do have a really large portfolio of antibodies that cover about two-thirds of the human proteome. We also offer immunoassays such as ELISA kits and IHC kits, and we also offer cell separation kits and antibody labeling kits. The company later expanded to include more product lines. So, for example, we also have our humankind branded cytokines and growth factors. So, all our proteins are expressed in human HEC293 cells, so you can be confident that they have the native folding, post-translational modifications, and glycosylation that you would expect to see in human proteins, and that ultimately increases their bioactivity. We also have our Chromatech branded nanobody based reagents. So, nanobodies are about 10 times smaller than traditional antibodies, and they can bind to their targets with really high affinity, which makes them ideal for various applications such as immunoprecipitation, immunofluorescence, specifically for super resolution microscopy, as well as protein purification. And then lastly, we also have a subsidiary of Proteintech known as Proteintech Genomics, and they manufacture antibodies that are conjugated to oligonucleotides for single cell RNA sequencing. So, as a company that is driven by scientists, we're here to support our researchers and scientific community any way that we can. So, our award winning products are self manufactured, and we offer the largest in house portfolio of antibodies for the human proteome. And then for convenience and testing, most of our antibodies are also offered in 20 microliter trial sizes. We also know the importance of product validation, and we offer open access validation data. Whenever we're testing new products, we validate with relevant sample types and using knockout, knockdown models whenever possible. Also, as antibody experts, we have in depth knowledge about antibody applications, and we can offer outstanding technical support. We also stay up to date with cutting edge technologies such as our proprietary antigen specific B cell cloning and engineering platform, which we use to manufacture our recombinant antibodies. Also, with 7 inventory sites and manufacturing and research sites on 3 continents, we are able to offer next day delivery to many areas of the world. Also, as a company, we do measure our success based off of our customer scientific success, specifically by helping them publish impactful research faster. So this graph here on the left shows that over our company history, our products are being featured in more and more publications each year. And as of now, we have over 300,000 publication citations, making us one of the top 5 cited antibody companies and also one of the fastest growing companies in product citations. I also want to highlight that as a company, we strive to make the searchability of our products on our website as user friendly and informative as possible. We've recently launched some new updates on our website to help our customers select the right antibody. This includes our unique 3D epitope viewer that allows you to interact with epitope structures and explore antibody binding sites so that you know exactly where our antibodies bind to your protein of interest. We've also introduced advanced filter options to help you find exactly what you need, such as our immunogen region selector toggle, which allows you to filter for antibodies generated from your region of interest. And then additionally, you no longer have to filter through all the variations of an antibody from the search results. You can view all formats of an antibody, including conjugations and buffer formulations, on just one page, which ultimately streamlines your process for antibody selection. So with that, I would like to go ahead and introduce our speakers for today. So today we have joining us Sophia Castelli, who is a PhD candidate at the University of Pennsylvania, Dr. Joanna Schaffel, who is a postdoc at Lausanne University and Ludwig Research Institute. Dr. Kirthana Ramanathan, who is a postdoc at the Technical University of Denmark. And our keynote speaker, Dr. Catherine Tomaro-Duchesneau, who is the Senior Director of Manufacturing and MS&T at RoslinCT. So we will give each speaker a short talk. So they will each give a talk, and then we'll follow that by a few questions from you guys, the audience. If you do have a question for one of the speakers, just please feel free to input that into the chat in the Q&A box. And if the speakers, do notice some questions that go unanswered live, they might respond through the chat function later. So definitely keep those coming, and just to make it easier, inform us who that question is for. So let's go ahead and get started with our first speaker. So our first speaker today will be Sophia Castelli, and her talk title is IL-9 redirects CAR T cell fate, enhancing efficacy against solid tumors. So Sophia, I will go ahead and pass it off to you.
Awesome. Thank you for that introduction. Go ahead and share my slides. All right, can you see my slides okay?
Yep, looks great.
All right, perfect. So yeah, I wanted to thank, first of all, Proteintech and Afrida and the rest of the organizers for inviting me to give this talk. Again, my name is Sophia Castelli, and I'm a PhD candidate in Dr. Carl Dunsab at the University of Pennsylvania. And today I'll be giving a short talk on my project, which is entitled IL-9 signaling enhances CAR T-cell anti-tumor efficacy in solid tumors by promoting memory phenotypes and reducing dysfunction under antigen stress. So although CAR T-cell therapies have been highly successful for the treatment of blood cancers, their efficacy for the treatment of solid tumors has been limited. And this is because of multiple challenges, including things like heterogeneous antigen expression, which can lead to immune escape, as well as the expression of tumor antigens in normal tissues, which can lead to on-target off-tumor toxicity. CAR T-cell expansion and persistence can also be limited after infusion. CAR T cells often have a limited ability to traffic to and penetrate into solid tumors, and the immunosuppressive solid tumor microenvironment can impair CAR T cell fitness. And so there's an unmet need to overcome these challenges for the treatment, of solid tumors. So a potential approach that we can use to improve the efficacy of CAR T cells for solid tumors is to engineer them to receive a cytokine signal. So full physiological activation of T cells requires three signals. The first one through the T cell receptor. The second one is through a co-stimulatory receptor. And the third signal is through a cytokine receptor. And this is required for optimal T cell proliferation, differentiation of naive T cells into effector cells, and the development of memory. However, CAR T cells that are currently in the clinic have been engineered to receive signal 1 through a CD3 zeta domain and signal 2 through a co-stimulatory domain, but they lack a genetic modification to provide them with signal 3. And so we can potentially engineer CAR T cells to receive this cytokine signal in order to improve their anti-tumor efficacy. And an important family of cytokines with pleiotropic functions within the immune system is the common gamma chain cytokine family. So this includes IL-2, IL-4, IL-7, IL-9, IL-15, and IL-21. And the receptors for these cytokines are formed by the common gamma chain, and then a private receptor chain that is unique to each of these receptors. And upon ligand binding, this leads to heterodimerization of these chains and downstream signaling, which is primarily through the STAT family of transcription factors, including STAT1, 3, 5, or 6, depending on the receptor. So in a previous study that we published in our lab in collaboration with the Ribas lab and the Garcia lab, we explored the therapeutic utility of providing CAR T cells with a signal from each one of these receptors from this family. And we found that when we provided CAR T cells with a signal from IL-9, this led to improved anti-tumor efficacy in preclinical models of solid tumors. However, the mechanisms by which this is happening are unknown. And so my project is building on this previous work, but trying to understand what are the mechanisms that are driving this improved anti-tumor efficacy. So to give you a short background on IL-9, it is produced by multiple cell types, including Th9 and Th2 cells, ILC2s, and mast cells. And it works on multiple cell types as well, including mast cells leading to their activation and secretion of mediators, B cells leading to memory B cell responses and IgE production, epithelial and smooth muscle cells leading to secretion of mucus and chemokine production, and eosinophils leading to inflammation and secretion of mediators. And so the roles played by IL-9 through these target cells include amplification of allergic reactions, as well as protective responses against parasitic infections. But with regards to the role of IL-9 signaling in T cells, it is not well characterized. It is known, for instance, that naive T cells are insensitive to IL-9 and that T cell development is not impaired in IL-9 deficient mice, suggesting that IL-9 is not critical for T cell biology. And importantly, in a recent study that was performed by Anusha Kalbasi's group at Stanford, they looked at IL-9 receptor expression across multiple tissues, and they found that IL-9 receptor expression is very restricted across immune cells, and in particular in T cells. Expression is very low. It is expressed in less than 1% of T cells, very rare subpopulations. And so basically the role of IL-9 signaling has not been well studied in IL-9, mostly because its receptor is not really expressed in T cells. So for my project, what we did was engineer T cells to express CAR molecule targeting the tumor antigen mesothelin, and this is expressed in multiple solid tumors, including pancreatic cancer, as well as the wild type IL-9 receptor, and then we termed these CAR IL-9R T cells. So first, to look into the anti-tumor efficacy of these cells, we used a xenograft pancreatic ductal adenocarcinoma, or PDA model, in which we implanted NSG mice with mesothelin-expressing PDA cells, ASPC1, by subcutaneous injection. And then after tumors were established, we treated them with CAR T cells expressing the IL-9 receptor or conventional CAR T cells by IV injection. And then to provide these cells with an IL-9 signal, we used an adenovirus encoding human IL-9, and we injected that intratumorally to achieve high concentrations of IL-9 locally within the tumor. So here are the results for overall survival, and you can see that when we treated with IL-9 signaling CAR T cells, so the combination of CAR IL-9R T cells and the adenovirus encoding human IL-9 shown in purple, we saw a significant improvement in overall survival compared with conventional CAR T cells. And importantly, IL-9 signaling CAR T cells are a lot more potent because they were able to have superior overall survival at a lower dose of 30,000 CAR T cells. And they were better compared with control CAR T cells at a higher dose of 1 million CAR T cells. And you can also see that quantified here in these tumor growth curves, where conventional CAR T cells at the 1 million dose led to complete remission in 2 out of 10 mice, whereas IL-9 signaling CAR T cells led to complete remission in 10 out of 10 mice. We also performed a tumor harvest in a separate experiment to look at tumor infiltrating CAR T cells. So first, we saw significantly more tumor infiltrating CAR T cells in the group treated with IL-9 signaling CAR T cells. These tumor infiltrating CAR T cells also had a significantly higher percentage of central memory T cells. And then these cells also had lower expression of exhaustion markers such as PD-1 and TIM3. And then to look into the phenotype of these cells in the context of repeated antigen stimulation, we performed an in vitro continuous antigen exposure, or CAE assay, in which we exposed these CAR IL-9R T cells to antigen expressing tumor cells for a long period of time. And so the samples that I'll show you for the following assays are either the CAR T cell product as a control, so this is day 0, the mock cells underwent the CAE assay in the absence of IL-9 treatment, and then IL-9, which underwent the CAE assay in the presence of IL-9 treatment. So first, we looked into tumor killing of ASPC1 cells, and we found that after the CAE assay, the cells that have been treated with IL-9, shown here in blue, led to significantly improved tumor killing in vitro compared with the mock-treated cells shown in green. And we also looked at interferon gamma production, and we found that IL-9 treated cells had significantly higher levels of interferon gamma production, both right after the CAE assay or after an additional stimulation with tumor cells. And so from this, we can conclude that IL-9 signaling is enhancing CAR T cell effector function through both improved tumor killing and interferon gamma production. We also looked into the phenotype of these cells by flow cytometry. And you can see first looking into the memory T cell subsets that cells after undergoing the CAE assay in the absence of IL-9 treatment, they have a decreased percentage of central memory cells, but IL-9 treatment leads to a shift towards the central memory phenotype. Also looking at the CD4 versus CD8 T cell percentage, we see that after the CAE assay, the mock cells have a lower percentage of CD4 compared with CD8 CAR T cells. But then after IL-9 treatment during the CAE assay, we see that this ratio between CD4 and CD8 T cells is preserved. And then finally, looking into surface CAR expression, we see that the mock-treated cells have a downregulation of surface CAR expression. However, when we treat these cells with IL-9, there's an increase in surface CAR expressing T cells. So to further look into what pathways might be driving this improved effector function and this phenotype, we performed single-cell RNA sequencing on these day 0 mock and IL-9 treated cells from 3 different donors. And here you can see the UMAP and the 15 different clusters that we were able to identify that correspond to different T cell subsets. And when we quantified the proportion of cells in each of these clusters for the different treatment groups, we found some significant differences between the mock and IL-9 treated groups. So first, the IL-9 treated groups had significantly more cells in the CD4 activated and strongly effector T cells compared with the mock group. And they also had significantly less T cells in the CD8 exhausted group. We also performed a trajectory inference analysis, and this allows to study dynamic changes in gene expression. So basically, it creates an axis of time or pseudotime, and it predicts which cells are closer to the beginning of the axis and which cells are closer to the end of the axis. And that way we can project the movement of cells over time. So we identified 4 different trajectories, of which today I'll show you 3. So the first trajectory is for CD8 T cells, and it goes from naive to central memory cells. And here we saw that in the IL-9 treated group, there were significantly more CAR T cells along this trajectory compared with the mock group, suggesting that IL-9 signaling is redirecting CD8 CAR T cells towards central memory clusters. And this also is consistent with the fact that we saw more central memory in tumor infiltrating CAR T cells, as well as after the CAE assay in vitro. Another trajectory that we identified goes from CD8 naive to CD8 exhausted cells. And in this case, there were significantly less cells along this trajectory for the IL-9 treated group, suggesting that IL-9 signaling is directing CD8 CAR T cells away from dysfunctional clusters. And finally, we identified one trajectory for CD4 cells, and this goes from CD4 naive to CD4 proliferating CAR T cells. And here we saw a difference in the distribution of cells along the pseudotime, where for the IL-9 treated group, we saw that significantly more cells are reaching this terminal endpoint of the trajectory, suggesting that IL-9 signaling is promoting progression towards proliferating states in CD4 positive CAR T cells. So in order to investigate the transcriptional mechanisms underlying these differences between mock and IL-9 treated cells under antigen stress, we performed a transcription factor analysis, and we found that IL-9 treatment significantly enriched STAT4 and STAT1 activity, both in CD8 and CD4 CAR T cells. So STAT1 is known as an established mediator of gamma chain or gamma chain cytokine signaling, including IL-9. However, STAT4 typically supports Th1 responses via IL-12 signaling, and it had not been previously associated with IL-9 signaling. So this was unexpected to see, and we're currently performing additional studies to verify if STAT4 activity is necessary to achieve this phenotype for IL-9 signaling CAR T cells. So as a summary, IL-9 signaling CAR T cells led to significantly improved overall survival in a xenograft PDA model, as well as increased numbers of tumor infiltrating CAR T cells with a central memory phenotype and decreased expression of exhaustion markers. IL-9 signaling CAR T cells have a unique phenotype after repeated antigen stimulation in vitro. So they had improved effector function, increased proportion of central memory T cells, a higher CD4 to CD8 T cell ratio, and higher surface CAR expression. And then we found that IL-9 signaling alters CAR T cell fates under antigen stress, potentially through STAT4 signaling. So it favors the transition of CD8 cells towards memory states and reduces their progression to dysfunctional states. And it promotes proliferation in CD4 T cells. So with that, I would like to thank everyone in the June lab, especially Carl and Gina, my mentors, as well as our collaborators, my thesis committee, and their Parker Institute for funding. And yeah, thank you all for being here today. And again, Proteintech for inviting me, and I'll take any questions. Thank you.
Awesome. Thank you so much, Sophia. That was a wonderful talk. We do have a couple questions there, both in regards to IL receptor expression. So the first question is asking, how does IL-9 receptor expression change during activation / differentiation of endogenous TCRs?
Yeah, so in our group, we haven't really looked that much at IL-9 receptor expression yet, but we're looking into that right now. But I believe that Anusha Kalbasi's group, they have their manuscript out in bioarchives, and they have looked more closely into IL-9 receptor expression. So I would probably refer to that paper.
Okay, so then I don't know if you'd have insights on the second question, which is also talking about receptor expression, but they're asking whether it decreases after central memory cells are formed in the CD8 CAR T cells.
Yeah, again, we haven't looked into that. It's also hard because the antibody that we found only it binds to the same spot where IL-9 binds in the receptor. So it's hard to detect once you've added the IL-9 because it won't bind. So we're looking into other strategies to follow IL-9 receptor expression, after activation and memory formation, but we haven't looked into that yet. But that's
Great,
a great
thank
point,
you.
though.
I think we have time for maybe one more question. I know there's a few questions coming in, and I'm sure Sophia would be happy to answer them via chat. But just one more question. Someone was asking what kind of analysis and software were used to obtain the cell clusters before the trajectory inference analysis. I think it was slide 11.
Yeah, so the analysis was performed by a bioinformatician in our lab, Dr. Wes Wilson. So he probably has a lot more insights into that, but also our manuscript is published in bioarchives as well. So I'm sure that's detailed in the methods, but I'm not a bioinformatician, so I'm not completely sure. Sorry.
No worries. Awesome. Well, thank you so much, Sophia. I think just for time's sake, we should move on to the next talk. So our next talk is by Joanna. So her talk title is Comprehensive Profiling and Dynamics of Tumor Reactive T Cells at Steady State and During Immunotherapy. So I will go ahead and pass it off to you.
Okay, can you see my slide?
Yep, looks great.
Okay, so thank you for the invitations, and I'm happy to be here today. So my name is Joanna, and I'm pleased to have the opportunity to present you today the main focus of my PhD project that I did at the University of Hospital in Lausanne, a beautiful city facing the lake and mountains in Switzerland. And it consists of analyzing the profile and dynamics of tumor-reactive T cells in cancer patients. So as we all know, As we all know, the treatment of cancer has largely evolved from surgery to radiotherapy and chemo, making up the three traditional pillars of cancer care. And to overcome resistance and lack of specificity, a fourth pillar was developed, immunotherapy, which rely on the patient's own immune system to fight tumors. So we know that there are different types of immunotherapy with the commonly used immune checkpoint blockades. But here for my talk, we are interested in cellular therapy. And as Sophia presented, there are two main subtypes of cellular therapy or adoptive cell transfer. So one is based on the fusion of genetically modified T cells or chimeric molecules called CAR. And the focus of this talk is on another form of ACD, which consists of the transfer of expanded tumor-infiltrated lymphocytes called the TILs. And these TILs are isolated from surgically removed tumor pieces, and they are grown in vitro in the presence of high-dose IL-2 before being reinfused to the patient who received prior chemotherapy. And we know that TIL therapy shows strong efficacy in melanoma patients. So with the recent analysis reporting 34 to 44% of objective response rate, depending on pure checkpoint blockade. And recently, a study also demonstrated its superiority over standard immune checkpoint blockade as second-line treatment. And so personalized TIL ACD is expanding to other solid tumors, but responses are less robust in these tumors. And this is likely due to the low abundance of tumor-specific T cells for solid tumors. And so to date, the following immune correlates of efficacy have been described. So first, tumor with high mutational burden as well as high neoantigen loads do respond better to cellular therapy. And tumor mutational burden has also been linked with the immunogenicity of the tumors and by extension, the infiltration of our TIL. So therefore, it's a common rationale to rather select patients with high infiltration status to obtain a rich product. And a key challenge for the success of this therapy is really to understand the mechanism of in vitro TIL expansion. So in this paper, they show in pancreatic cancer and melanoma patients that dominant tumor-infiltrating clones were outcompeted by newly emerging clones, and poorly expanded TILs show sign of exhaustion. So the dynamics finally impacts the final cell products. And it has been shown that the quantity of the TIL infused in the patient, as well as their tumor reactivity, correlate with better clinical response. And also the diversity and frequency of neoantigen-specific T cells in the product are linked with better outcomes. And lastly, the cell state matters. So it has been shown that less differentiated stem-like cells lacking, for example, CD39 and CD69 drive response to TIL ACD. And finally, many studies also have linked the persistence of the TIL post-infusion to clinical response. And this includes not only bulk TIL, but also tumor-reactive and neoantigen-specific clones whose sustained presence correlate with in vivo anti-tumor activity. So in this context, some questions remain unanswered on which we try to focus during this project. So first, we wanted to analyze the cell types and their interaction in the tumor microenvironment of patients. We also dissected the dynamics of our TILs. So for that, we use TCR sequence as a single molecular barcode to track our cell from the baseline tumors to the product and to post-ACD samples we had access to, so a blood and a tumor biopsy obtained 30 days after infusion. And this allowed to understand the mechanism of TIL expansion as well as their in vivo fates. And then we also characterized the efficacy of our product, and we also could annotate some tumor-specific TCRs and track them in our pre- and post-ACD samples. And finally, we also investigated how TIL adapts post-ACD to antigen rechallenge. And so to answer all these questions, we took advantage of a clinical trial launched at the University Hospital of Lausanne under the supervision of Professor Koukos on 13 patients with melanoma that received TIL ACD. And what we see is that at 3 months, we observe a partial response for 6 patients, out of which 2 are now complete responders, and 7 patients that did not respond to the therapy, so 3 with stable disease and 4 showing progressive disease at 3 months. And just rapidly, the main technology we use for the analysis, so we used an in-house method of bulk TCR sequencing called CTR that allows efficient TCR identification, and we show its superiority in resolution depth and reproducibility over the leader immune sequencing provider Adaptive Biotech. And also, for sure, I used extensively single-cell RNA-seq from 10x Genomics, both on the baseline tumors and on the product to understand the different expansion and in vivo dynamics. And so first, we wanted to decipher the main cell types and their interaction in baseline tumors. So we profile our baseline tumors by single-cell RNA-seq, and we identify about 90,000 cells. And when we focused on the malignant compartment, we observed that responding tumors have higher immunogenic programs, including interferon responses, antigen presentation, and immune checkpoint expression. And we then enriched the immune compartments by sorting for CD45 expressing cells. And we look at the myeloid compartment, and we found distinct clusters of macrophages and DC. We didn't find any difference in terms of proportion by response, but what we observed is that responders show activation signaling in DC in macrophages like interferon and antigen presentation signatures. And then we also wondered if this activated myeloid profile correlate with greater T cell infiltration, and it did. So responders' tumor were enriched in TIL. And then we focused on our TIL, so mainly on the CD8s. We observed 9 subtypes. And what we saw is that in responders' tumor, precursor exhausted and terminally exhausted subsets were enriched, and these subsets are subsets that are associated with tumor-specific cells. And so this suggests that effective TIL-ACD is characterized by cellular crosstalk between our TILs and myeloid cells. And by performing ligandome analysis, we could really reveal more immune cell-to-cell interaction in responders' tumors, and this mainly implicated T cells and myeloid cells. And so we think that this dense niche likely recruit and nurture tumor-reactive TILs that are then the basis of effective cellular therapy. And so then we dissected the dynamics of our TIL. So as I told you, we use the TCR as a unique barcode to track specific clonotypes. And we looked first at the overlap, thus the common TCR of repertoire between baseline tumors and our cell products. And looking at the fraction of tumor repertoire that expanded in product, we observed this fraction to be higher in responders. And when we annotated our repertoire with single-cell data, we saw that it is the CD8 compartment that is preferentially mobilized in tumors from responders. And then we looked this time at the fraction of the product that originated from the tumors, and unexpectedly, we observed a significantly higher fraction in responders' products. So we wonder where the clones from non-responders were coming from, and so we looked at the overlap of the products, this time with the blood repertoire obtained at the same time than the surgery. And we observed that, in fact, non-responders have product enriched in blood-borne CD4 clones, while responders have product enriched in so-called tumor-resident cells, which are tumor-derived T cell clones. And then we wanted to understand the mechanism driving this greater in vitro expansion of responders' TILs. So we hypothesized that their in situ frequency could drive their propensity to expand. And so this graph is a bit hard to understand, but I will try to guide you through. So when we normalize the tumor repertoire into 5 quintiles of equivalent proportion of TCR, we observed that the most dominant TCRs here in blue were more prone to expand than the lower frequency one. And this is summarized here in this heat map. So you see the impact of the frequency on the propensity to expand, but still we can observe a significant difference between responders and non-responders, suggesting another mechanism than the frequency. And thus we wonder if this could be linked to the state of our cells. So we went back to our single cell data, and we looked this time at the proportion of clone mobilized in cell product within the different CD8 subtypes. And we see that there is a tendency for precursor exhausted cells in light purple to preferentially expand, but the expansion capacity is pretty heterogeneous among the different states and among patients. But of interest, when we look at the 2 exhausted populations that are enriched in our responders, we observe that exhausted cells from responders do better expand than these of non-responders, suggesting underlying mechanism. And finally, as a summary, this ends up in responders' product being enriched in tumor-resident CD8 cells that are mainly composed of exhausted cells, while the few tumor-resident clones you found in non-responder products stemmed mainly from effector memory-like cells. And then we wanted to understand the mechanism of their in vivo fate once infused into the patient. And so we did the same kind of analysis, looking this time at the product and a tumor biopsy post-ACD. And given the large fraction of blood-borne clones in non-responder product, we analyzed their in vivo fates based on their frequency in the product, but as well based on their origin. So what we can see is that by dividing the product repertoire into 5 quintiles for each tumor-derived and blood-borne fraction, we see that tumor-derived clonotypes effectively infiltrated tumors post-ACD in patients who experience objective response or stable disease, while they were rather confined to the circulation in patients with progressive disease. And conversely, when we look at the blood-borne clonotypes, they repopulate mostly blood and infiltrated tumors in non-responders, mostly because they were at super high frequency in their products. And then we focused on the product itself. So we know now that the product from our responders have a higher number of tumor-derived TILs. And we wonder if these TILs are tumor reactive. So we co-culture our TILs with autologous tumor cell lines and look at the tumor reactive fraction by CD137 upregulation. And as hypothesized, responder product contain a higher proportion of CD137 expressing CD8 cells that translate into a larger absolute number of infused tumor reactive cells in this patient. And to validate these findings, we then selected some dominant TCR, and we cloned them into recipient cells and screened them against tumors. And we observed that indeed product from responders have a larger proportion of tumor reactive that translate into a higher fraction of their products. We then also assessed tumor reactivity for some of the dominant clones from our baseline tumors. And we find also that tumors from responders are enriched in tumor reactive CD8 clones. And finally, we ended up with over 240 annotated clonotypes. And with that and our single cell data, we could build a signature of tumor reactivity. And so we then used our signature and we predicted that predicted tumor reactive clones are enriched in exhausted cluster, as it is known in the literature. And we can also see that our infused products show greater TCR diversity and higher number of inferred tumor reactive clones for responder patients. So to sum up and illustrate these dynamics of tumor reactive TILs, so you see that responders have tumor enriched in the number and fraction of predicted tumor reactive clones. A third of these clones then expand in their products, making up a quarter of their product repertoire, while in non-responders, only 25% of the predicted tumor reactive clones from the tumor expand, and only a third of them then are able to engraft tumor post-ACD, while in responders, half of the predicted tumor reactive clones from the product are able to infiltrate the tumor post-ACD. And since the efficacy of the product has been linked with the less differentiated and stem-like phenotype of expanded TILs, we also wanted to see what was the state of our cells in our product. So we had performed single-cell RNA-seq on the product, and we see that our cognate cell products are not clustering with the canonical tumor states, and that the clustering was in fact strongly driven by individual patients. And what we observed is that the discriminant gene of in situ canonical states are completely anonymized in cognate products. And we see in particular downregulation of exhaustion genes, and products are rather characterized by effector and cytotoxic genes. And then we wanted to understand how engrafted TILs adapt to in vivo rechallenge and cues from the TME. So we also had performed single-cell RNA-seq on some of the post-ACD biopsy, and we see the different transcriptional changes from the baseline tumor to the product and to post-ACD tumors. And we see that the major transcriptional changes occurs in responders from T0 to product, indicating this reprogramming away from exhaustion. The states are more stable from the product to post-ACD biopsy, but still more dynamic for responders. And when we did some pathway analysis, we see that TCR and PD-1 signaling are downregulated after expansion, and they are reactivated post-infusion, specifically in responders. And so to summarize, we see that cytotoxicity is high in products, but lost in non-responders post-ACD biopsy, while exhaustion and PD-1 signaling completely lost during expansion rebound in responders after infusion, suggesting really an active immune response supported by re-established myeloid crosstalk in this patient. And finally, to conclude, we identified different immune correlates of TIL-ACD efficacy, mainly within baseline tumors of responding patients that are enriched in activated myeloid cells that recruit exhausted tumor-reactive cells. These cells are then capable to expand, and it generates big cell product with high tumor reactivity, while in non-responders, the absence of these cells leads to the outgrowth of circulating CD4 T cells in the product. And we calculated that on average, responders received 50 billions of tumor-derived TILs, including 32 billions of them predicted as tumor-reactive, while non-responders received only 40 billions of these tumor-derived TILs, with less than a billion of them predicted as tumor-reactive. And we also demonstrated the reprogramming that cells encounter during expansion. And finally, tumor-derived clones preferentially ring-graft in post-ACD biopsy of responders. And they re-upregulate exhaustion markers, really showing a sign of active immune response. And so with that, I want to thank all people and former members of the Harari's group, as well as the Dengach team with whom I collaborated closely. And a particular thank to my PhD mentor, Professor Harari, and co-supervisor Kukos and Denarda Dengaj, and you for your attention. Thank you.
Amazing. Thank you so much, Joanna, for that wonderful talk. We do have one question in the Q&A box asking, you showed that TILs can decrease exhaustion during in vitro expansion. Does the same occur with homeostatic proliferation of TILs?
No. So this is really because we are in a setting that is artificial. So we still have a bit of cues from the TME at the beginning, because what is happening during TIL expansion in the lab, so you take a piece of the tumor and then you have them in a petri dish and you make them expand. So it's a bit artificial, and you give also a lot of IL-2 doses at a really high dose that is not the dose that you have homeostatically. So it really changes the way your TIL are expanded, and this is why we see all this reprogramming that we wouldn't see otherwise.
Okay, wonderful. Thank you so much for clarifying and explaining that. So just for time, I think we should move on to our next speaker. If you do have any questions for Joanna, feel free to continue putting those into the chat or Q&A box, and she can answer them there. So our next speaker is going to be Kirthana. Her talk title is Generation of Efficacious Genetically Engineered CAR and TCR T Cells Using Antigen-Specific Nanoscaffold Media.
Hello, can you hear me? I hope everyone can hear me. Frida?
Yeah,
Yep.
we can hear you.
Perfect. I just went silent for myself for a moment. I don't know why. All right, so my presentation today is on generation of efficacious CAR or TCR T cells using scaffold-mediated expansion. And I would like to thank Proteintech for giving me this opportunity to present my project. This is one of the early stage projects, so I will just present the data I have for now. Hope we can have some fun with it. All right. So adoptive T cell therapy has been around for quite a while, from 1980s. One of the first adoptive T cell therapies we had is tumor infiltrating lymphocyte therapy, as Joanna just explained. And recently we have developments on CAR and TCR engineered T cell therapies. In TCR engineered T cell therapies, usually you have a TCR that has already been identified, and these are engineered into the patient T cells, expanded and infused back into the patient ex vivo. Similarly, in the CAR T cell therapy, we use a different receptor called chimeric antigen receptor, as you all know. And this is engineered into the T cells, expanded and infused into the patients. And these, both TCRs and CARs, they target the tumor cells. So here we are going to just focus on the receptor engineered T cell therapy. The expansion protocol of this receptor engineered T cell therapy clinically is quite exhaustive. So the process involves taking out human T cells from the patient, activating them with CD3, CD28, which is signal 1, signal 2, required for T cell activation and expansion. And this can be done through several ways. So one of the easiest ways is to use soluble antibodies of CD3, CD28, but we also have commercially available and artificial antigen presenting platforms such as Transact and Dynabeats that also has the CD3, CD28 to activate these T cells. Once these T cells are activated, the chromosome opens up because they are up to proliferate. So this stage is what we use or leverage to engineer these T cells. So we usually use a retro or lentiviral vectors that are carrying CAR or TCR gene to insert this CAR or TCR gene into the T cells. But recently, there has also been development in using CRISPR-Cas9 mediated knock-ins, where we use double-stranded templates or circular templates or single-stranded templates that codes for CAR or a TCR, and we use the Cas9 mediated knock-in into the T cells. Once we produce these T cells using this engineering, you can take these CAR or TCR T cells and expand them to clinically relevant numbers, which is usually more than 100 million at least. So to expand them to clinically relevant numbers, you need to expose them to quite high concentrations of IL-7, IL-15, or IL-2, or usually we can also use a combination of several cytokines, for example, IL-2, 715. Once these are expanded, they are infused back into the patients so these T cells can traffic to the tumors and take them down. And it's been known for a while, and it's also been shown in this very nice study in 2020 in Nature Medicine that the clinical efficacy of CAR T cells, similarly TCR T cells, are also linked directly to the T cell phenotype. So when you have more memory CD8 T cells, you have a complete response. And when you have more exhausted CD8 T cells, you have progressive disease or partial response. And this is mainly because T cell phenotype changes during the activation and expansion phase of these T cells. A naive T cell, which has not received any activation or expansion, has their most self-renewable capacity or proliferative capacity, which means they persist the most. And once you activate them and expand them, they go through several phases of differentiation, which reduces their self-renewable capacity, but increases their cytotoxic capacity. But at the end stage of their differentiation, which is the terminally differentiated T cells, they have they are very plausible to have higher exhaustion, but also they can go through cell death and senescence quite easily. So when you have an exhaustive expansion protocol, then it's quite often we have terminally differentiated T cells in the final expanded cell product. So if you have to polarize these T cells to have more memory-like phenotype, which is like central memory or effector memory, you could use several engineering techniques like Sophia introduced, but you could also target different parts of the production protocol so that you can change the way you activate the T cells and receive a better phenotype at the end. And the most important parts where you can target during the production protocol, or the first step where you activate these T cells for engineering, or the last step where you expand them with really high concentrations of the cytokines. And one of the ways to do it is using artificial antigen presenting platforms. And these artificial antigen presenting platforms involves biomaterials such as polymers or dextrans. And now, most recently, there has been a lot of development in lipid nanoparticles that can also provide the same type of cues that a dendritic cell could provide in vivo. So here in this project, we use a dextran-based scaffold, which is a linear dextran molecule, which has streptavidins embedded in it. So we can click on an antigen. So we get an antigen-specific activation while you expand these T cells. But at the same time, we could also click on either cytokines or co-stimulatory molecules so that the antigen-specific activation of this T cell is combined with paracrine delivery of the co-stimulation or the cytokines. So as I explained, Dextran is a linear molecule. What we use here is embedded with streptavidins, and as long as you have your antigen or cytokine or co-stimulatory molecule monobiotin related, you can click them on through streptavidin biotin activation. So once you click them on, you can culture the T cells and you can expand them antigen specifically. This is highly customizable. Of course, Dextran is just a sugar molecule. It's biocompatible, and you can also make sure you have an antigen-specific activation of these T cells. So we have done this before, and it's been published in the context of viral-specific T cell expansion, or in terms of melanoma patients, we have done TIL specific T cell expansion. And here we show that when you have a PMHC, which means a peptide clicked onto the MHC with co-stimulations and cytokines, we can expand in a PBMC from somewhere between 0.7% viral-specific T cell all the way up to 80% or more. And this is what I show here is either CMV or EBV specific T cells. And you can go into this publication to read more about it. So we wanted to leverage this technology. And in this paper, you will also see the scaffold-mediated expansion will also have a favorable phenotype when they're expanded. So we wanted to leverage this and use it on CRISPR-Cas9-mediated CAR T cell therapy. So it has been shown in 2017 by Michel Sadelain and Justin Ekum that if you knock in a CAR in the track locus of the TCR, then these CAR T cells that are produced from them have better tumor killing efficacy, also have less exhaustion. So we wanted to start here because when you do knock-in without using a virus, we have very low knock-in efficacies, especially when you use a cheaper method like double-stranded DNA and just the natural Cas9. We have very low knock-in efficacies. So we thought we could use the scaffolds to expand them into clinically relevant numbers. So what we did was on day 0, we activated these T cells. We used a double-stranded template with an RMP, and we knocked them in on T cells. For TCRs, we had a double-stranded template of the TCR of interest. Here we used NY-ESO. And for the CAR, we had the CD19 CAR. So when we knocked in, as you can see here, we could get somewhere between 5 to 10% in the CAR knock-ins, and we could get somewhere between 10 to 20% in the TCR knock-in, which is quite low and it's not clinically relevant. So we decided to feed them with the scaffolds, and here we used TCR or CAR antigen along with IL-2 or IL-21 on the scaffold. And after the knock-in, we rested them for 48 hours, and then we started feeding them with the scaffolds on day 4, day 7, and day 10, and we harvested them on day 14 for analysis. So with the TCR NY-ESO, when we expanded them just with IL-2 715, which is a cytokine cocktail, non-specific expansion, we had about the same percentage in the total pool of CD8 T cells, which we received on knock-in, so 20% roughly. And when we expanded them with scaffolds on day 14, we could see almost 80 to 90% specific T cells. And with the CAR, quite similar. However, we had a little bit less expansion, but we still could reach up to 50% specific cells, which is quite high when compared to what we start with, and the cytokine-mediated expansions does not achieve high numbers. And if we look at the phenotype, just for the time's sake, I'm showing one of the most relevant differences, which is CD27-28 double positive cells, which has higher self-renewable capacity, also is good effector memory capacity. And the scaffold expanded T cells are enriched in this population in both CARs and TCRs. So basically, we know we can do the expansion specifically, but we needed to know if it improves the functionality of these T cells. So we went on to look at the proliferative capacity of the T cells. Here I show the CAR data, CD19 CAR data, just because of time, but we have this data for TCRs, both lentivirus and CRISPR CARs. So if you see, what we did was to take the T cells, CAR T cells after expansion, treat them with CFSE dye, let them expand with irradiated CD19 target cells. Here we use JACO1 B-cell lymphoma target cells. And after expansion, for 6 days, we looked at them in flow cytometry. then you can see the dilution of the CFSC. And we could see that the antigen scaffold expanded T cells, CAR T cells, they had the highest proliferative capacity. And we also had, in comparison, two types of cytokine-mediated expansion, IL-2 715 and IL-715, which showed much less proliferative capacity. And we also looked at one of the antigen-specific expansion protocols where we used JCO-irradiated target cells with IL-2 and IL-21, the same cytokines that were on the scaffold. And then we could see we can rescue a little bit of the proliferative capacity, which means the antigen specificity of these scaffolds actually promotes this kind of higher proliferative capacity. And then we went into a rechallenge killing assay in vitro, a cytotoxic assay we performed with InQSite, where we seeded 1-to-1 affected targets, CD19 CAR against JCO1 loop tumors, and we rechallenged these CAR T cells every 72 hours. And when we did that, over 4 rechallenges, we could see the antigen scaffold expanded CAR T cells could eradicate the tumor throughout the assay. And there was some killing until the second rechallenge with the IL-715 expanded CAR T cells and also the JCO-irradiated expanded CAR T cells. However, after the second rechallenge, all of them lose the capacity to kill tumor and they have relapse. So this motivated us to look at how these CAR T cells evolve during when they see the target cell. Something happens when they see the target cells, and that's also why we lose their efficacy. So when we look at them during the rechallenge, we could see with the interferon gamma expression of these T cells, the antigen scaffold expanded CAR T cells express interferon gamma throughout the 4 challenges in quite high amounts. And if we look at the CAR T cells after the expansion, the central memory cohort of the antigen scaffold expanded CAR T cells are enriched. And they also have lower number of TEMRA or terminally differentiated CAR T cells. So further, we went into a mice experiment where we injected the mice with 1 million JCO cells, which is a B-cell lymphoma, contains the CD19 target. We let the tumor establish for 7 days, and we did a dose de-escalation test where we did 2 different doses of CAR T cells, 0.5 million and 0.25 million CAR T cells. They are either expanded with IL-2 715 cytokine cocktail, or they're expanded with antigen scaffolds. And we scanned these cells, scanned these mice on day 14, day 21, 26, and took them down on day 33. And throughout the experiment, we could see that the antigen scaffold expanded CAR T cells in both the conditions completely eradicated the tumor, did not allow any tumor growth until day 33. However, when we had the cytokine expanded CAR T cells, we had a tumor escape already in day 14 in the 0.25 million condition, and in day 21 in the 0.5 million condition. We had to take down all of the mice in 0.25 million condition on day 21. However, the mice survived for 0.5 million in the IL-2 715 condition, but they had incredibly high tumor burden. So this to conclude, we can see that the CRISPR-Cas9 mediated knock-in of CAR or TCR can give you 5 to 20%, depends on your technique. And we can use the scaffold to further highly enrich these T cell products. And these scaffold expanded CAR and TCR T cells have a high self-renewable phenotype, high proliferative capacity, and thereby have a superior in vivo and in vitro cytotoxicity. So as an epilogue, it is beneficial to use antigen-specific mediated expansions for these kind of CAR or TCR engineered T cells, and using artificial antigen presenting platforms like scaffolds could really improve the final clinical efficacy of this therapy. So with that, I would like to thank my group. I would like to thank Sine, who is the main supervisor of this project, and Maria. And I would also like to thank all the mice that died. That's it.
The forgotten, sacrifice. So thank you so much. That was a wonderful talk, Kirtana. We do have a question that I think we have time for. So Someone's asking, they're very curious on the artificial antigen presentation platform. How does the manufacturer platform look like in terms of scalability and repeatability from batch to batch?
That's an excellent question. We already had this platform in a phase 1 trial for melanoma patients, so we can scale them up enough for a clinical setting. And this is basically assembled by yourself. So we order a controlled extend batch on which we can also order GMP cytokines, co-stimulations, or antigens. So we click them on ourselves in the lab. So it is quite flexible and easy. However, for GMP production, we had to do more of a, how do I explain, more of a mechanical method where we used a lot of robots basically to do the same. But it
Very
is
interesting.
possible to scale, and it is repeatable. The repeatability is pretty good. Yeah.
Very interesting. I think we have time for maybe one more question before we
Mhm.
should move on. The second question is, what is the mechanism of the scaffold activation and how is it different from activation with individual cytokines?
So the mechanism, as we have seen through fluorescent microscopes, that these scaffolds, they stick on to a particular cell. They are really small. They are less than 20 nanometers. So they can sit on the same cell where they provide the antigen-specific activation and the cytokines to the same cell at the same time. So it's more of a paracrine-mediated activation. So that is why we are able to enrich them, enrich only the antigen-specific cells. But in the soluble cytokine setup, you will provide the stimulation to all the cells basically in the pool, reaching a polyclonal expansion, which leads to expansion of also the non-specific cells in high numbers, which might
Great.
not be as beneficial.
Perfect.
Yeah.
Thank you so much for explaining that, Kirtana. If you guys do have any additional questions, feel free to put those in chat or Q&A, and I'm sure she'd be happy to answer. So with that, we are ready to move on to our keynote speaker. So Catherine's title for her talk is Translating Potential CDMO Perspectives on Bridging Discovery and Scalable Cell Therapy Manufacturing. So Catherine, I will go ahead and pass it off to you.
Awesome. I'm Catherine, Senior Director of Manufacturing and MSAT at RoslinCT, the Boston site. We're going to transition a little bit and talk about CDMOs and their role in discovery and scalable cell therapy manufacturing, including transition from the lab and the bench to large-scale manufacturing and supporting patients. So I don't have to spend a ton of time on why cell therapy is important, but definitely want to put some faces to what it is that we do every day, both in development and in manufacturing. And if you haven't read the stories of Emily Whitehead or Victoria Gray or Lori Adami, I definitely encourage you to read a little bit. Emily Whitehead, the first pediatric CAR T patient. Victoria Gray had the first what is now Casgevy and for sickle cell anemia, and Lori Adami, who had tremendous recovery following a CAR T treatment of what is now Yescarta. Cell therapy does have still some challenges, both in development and in the manufacturing space, though. So we're going to talk about these, and they include things like complex and costly processes with limited proven scalability. A lot of the work is still in the early development stage too, including in academic centers with limited compliance and translational experience that is needed to support agency requirements. Most of the work is still in few full-scale runs, and so things like critical process parameters and critical quality attributes have yet to be defined. There still remains a bit of a lack of standardization across unit operations. So if you're performing an enrichment of cells, what technology are you using? If you're doing fill and finish, what technology are you using? It varies across labs and it varies across manufacturers as well. We can also talk about highly variable starting materials. So especially in the autologous space where we're doing healthy donor runs, how does that translate over once we start doing actual patient manufacturing? So one added challenge too starts to arise when we start trying to move a product into the clinic. So the translation to the clinic requires a much stronger documentation, process rigor, and compliance. And typically the scientists who did amazing work on the development side don't have the GMP training nor the understanding of the regulations that is needed to bring a product to the clinic. There's also now a need for an ability to scale up and scale out manufacturing while maintaining product quality and efficacy. So we're no longer doing the 1, 2, or 3 replicates that we would do in a lab setting, but are being asked to maintain reproducibility across thousands of batches. A lot of the processes also still have frequent manual and open steps, which require strict aseptic controls and a clean room infrastructure that's just not present in your typical lab environment. Moving to the clinic with the agencies also requires stringent requirements on the regulatory side that really aren't typically supported in that small scale setting. Working with cells obviously brings another challenge on its own, which is the need for intricate logistics, right? We're dealing with short vein to vein timelines with limited product stability. So there's a need for rapid testing and disposition. And all these challenges need to be overcome with limited time and money. And so how do we then transition things like the cell therapy programs from a development space, like an academic research lab or a biotech, into the bedside? And what I really want to drive across today is that a CDMO can help. Now, I know when I was in grad school and postdoc, I had never heard of what a CDMO is. So what is it? We can look at the acronym. So it is a contract development and manufacturing organization. And I want to stress that it is both the D and the M. So we're often known as manufacturing organizations, but we do have development space as well. And so the goal of a CDMO is to operate through development, tech transfer, manufacturing, analytics, and more. So provide a skilled and compliant end-to-end solutions, really with the goal of accelerating the path to clinic and commercialization. So with that, I want to talk to you a little bit about my personal journey to cell therapy CDMO and how I got here. So I started in the academic path, like most scientists do. I was at McGill University up in Montreal, so my hometown, and did a bachelor's in microbiology and immunology. I was able to acquire an industrial scholarship early on in my bachelor's, and so was able to collaborate with a small startup company and had the ability to do not only R&D like most bachelor's levels students would have, but I was able to participate in preclinical studies and also clinical manufacturing and quality control. And so that really gave me kind of a path forward towards more of the translational science. And so I continued and proceeded to a PhD in biomedical engineering, still at McGill University and still with the same startup that I had started with during my undergrad, and did this PhD with an industrial fellowship. So I got to continue some of the work I was doing, including helping run a clinical trial in that small startup. After PhD, and I decided I wanted to continue along the path of what I was doing and took the natural step to a postdoc and started at Michigan State University, and quickly 6 months in or so, my PI got recruited to Baylor College of Medicine at the Texas Medical Center in Houston. And if you haven't been to the Texas Medical Center, definitely recommend the visit. It's a pretty strong campus for collaboration and driving science forward. And so while I was there, I was actually working under an industrial grant for one of the big pharmaceutical companies, doing some development and preclinical studies. Actually, interestingly enough, it was quite focused on GLP-1 at the time, which many of you know now has been important in the development of Ozempic and other drug loss therapeutics. And actually at Baylor, with the partnership with this pharma company, I actually generated a patent, but it opened my eyes as to, okay, so what's next? I have preclinical efficacy data. How do I transition that to patients and actually make this a drug development space? So I didn't really know how that worked or how to even get there, but I did take another role. And so after academia, I transitioned over to industry with my first position as a technology transfer scientist, and then shortly after, director of manufacturing at small biotech company in Minnesota. So we were doing iPSC line generation and banking and some differentiated iPSC-derived products for drug discovery and toxicology with some of the big pharmaceutical companies. Come March 2020, the pandemic hit. Obviously not great for when you're working in a small biotech company with what was happening with the funding situations and the shutdown of pharmaceutical labs. And not only that, but on a personal level, my mom got diagnosed with stage 4 carcinoma. And so taking those things together, I made a career decision to move to actually Memphis, Tennessee at that point to take a leadership role in manufacturing for cell therapy CDMO, which brought me much, much closer to the patient. And I was able to lead the team doing not only work on cancer treatments and autoimmune diseases, but also got to contribute to COVID therapy development for a Treg program to try to help immune regulation in COVID patients. So it really allowed me to kind of feel closer to the patient, which brought quite a bit of personal gain as much as the focus was really on the patients. And now I am in Boston at a different cell therapy CDMO. And I will say in the 5-ish plus years I've been working in the cell therapy CDMO space, I've had the opportunity to work on over 30 cell therapy programs, anywhere from phase 1 to commercial manufacturing. So it's been a very rewarding experience thus far. And so where I'm at right now is RoslinCT, which is a cell therapy CDMO that has transatlantic development and manufacturing capabilities. We do have 2 sites. The first is in Edinburgh, Scotland, and is one of the manufacturers of the Casgevy program. You might know RoslinCT from the Roslin Institute, where the work on Dolly the sheep happened. So Roslin is definitely known for embryonic stem cell development and now iPSC development. The site I'm located at is Hopkinton, Massachusetts, which is also has multiple cell therapy programs, but is also partnering with Gameta for their commercial cell therapy manufacturing. If you don't know where Hopkinton, Massachusetts is, it's actually right outside Boston, and it's known for being the Boston Marathon start line. So the thing about CDMOs, and Roslin in particular, right, I'm a little biased, obviously working at Roslin, but is that we share a mission and vision that goes closely with product developers. We just do it a little bit differently, right? But at the end of the day, our goal is to save the lives of patients. And we do that by partnering with innovators and early product developers to support those transitions through the clinical phases and hopefully commercial manufacturing so that we can deliver on-time product to patients. And so why use a CDMO? And really this is what the entire goal of this, right, is to leverage existing CDMO infrastructure. So CDMOs are well known for their GMP ready clean room suite. So basically clean rooms that allow aseptic processing, so have low particulate and low microorganism levels. We have a ton of skill set in making sure that these clean rooms are up and running to the agency required levels. This includes equipment that's validated and ready for use, suites that are constantly monitored and maintained and have been through regulatory inspections. CDMOs also provide environmental monitoring and compliance systems, which include EMA or FDA or whatever other region you're operating in, which has pretty strict controls for things like contamination, right? Cell therapy is not sterile processing, it's aseptic processing. So we want to make sure the controls are really in place there. There's also extensive raw material and inventory management, which includes supplier qualification, raw material testing. When you're in the lab sometimes and you pick up a material and it's cracked or it doesn't work, that's not something that we can do when we're working with an autologous product in our hands where you really have a right first time requirement. CDMOs also have very established disposition systems, which also include QC laboratories, so we can ensure timely batch disposition and a short vein to vein time. So really by leveraging a CDMO, you can reduce the time and reduce the cost it takes to get to the clinic and deliver multiple batches of high quality. CDMOs also have staff that are experienced, right? So they go through extensive training programs, which include technical operations, clean room behaviors, aseptic practice, and compliance. They're really trained with a comprehensive qualification program to ensure right first time. Again, when you're working in an autologous cell therapy space, you typically got one shot on goal. So we really want to make sure that they're well trained in this space. Our operators typically have decades of experience and experience across multiple modalities and cell types, including process instrumentation. Their goal is really to deliver an efficacy for the product, but also with a focus on patient safety and with compliance to make sure that we're following all the regulatory systems. CDMOs provide a ton of technical skill set, and really it's learned from experience, right? We talked about in my personal career development that in 5 years I've touched over 30 programs. So I have a lot of learnings across different cell types and across different products, anywhere from CAR-T to TCR to iPSCs, speaking autologous and allogeneic, small and large scale. We also have quite a bit of experience on the process, regulatory, and analytical side that spans development phases. So we've been skilled to understand how phase 1 requirements are different from a phase 3 or different from a commercial manufacturing process. So we have deep understanding of the GMP requirements and things like comparability and validation. We've also had the ability to have extensive agency interaction experience, example with the FDA and the EMA. And so we also maintain strong partnerships with technology providers to make sure we're staying on that edge of innovation, but also being aware if there's any process instrumentation issues that we need to know. So really, as a CDMO, we aim to become a technical extension of the partner and be deeply involved in the success of the program. And really, 2 brains are better than 1, right? If we can utilize the process developer's technical knowledge of the process while providing some insight, both from a technical level, but also a compliant level, it's really driving that program to success. We also aim to provide experience and knowledge from product development all the way to logistics and regulatory. We aim to understand the science, but also have teams built with a strong technical skill set for which you can trust your program. We really do also want to provide personalized technical support and really make sure that it goes beyond the please execute 100 batches, but also goes to, hey, I think maybe you might want to do this way because A, B, and C. So with that, where does one even begin? And really, there's an entire other process that we could spend a lot of time talking about, and that's technology transfer. But I went ahead and grabbed the FDA definition of it, which is the transfer of product and process knowledge between development and manufacturing. Now, this could happen within one organization where technology is moving along from R&D to development to manufacturing, but it also is key when you're a product developer now giving your process knowledge over to a CDMO. And when I introduced myself, I said that I've had leadership roles and currently am in a leadership role that not only covers manufacturing, but covers MSAT. And so MSAT, which stands for Manufacturing Science and Technology, is really a product steward at the CDMO when it comes to technology transfer. So they're a technical team, often with a product development background that comes with a mindset for GMP manufacturing operations. So their role is really to drive technology transfer from the partner to the CDMO. They ensure robust and scalable processes across clinical phases and commercial. They do things like supporting process characterization, process trends, risk mitigations, and continuous improvement. So they're really just making sure that all the pieces that need to happen during tech transfer are happening at the right stages. And so are kind of the translational folks between development and manufacturing. So some things to consider as you look at a CDMO and consider tech transfer. Involve your CDMO early, even when you're still in development. One of the things we highly recommend with our partners is doing a GMP gap analysis. So looking at your development work holistically and seeing where could be the compliance gaps, where could be the analytics gaps, or maybe the supply chain gaps. That could look like performing a risk assessment as well, just looking at the critical steps in your process that might need some tweaking before we even consider GMP manufacturing. I also always recommend to invest in process and analytical development early. It's really easy sometimes to say, oh, we'll handle that when we're in phase 3, but then you're entering a comparability space from a compliance perspective that just becomes a lot more challenging. And so also definitely consider a transfer to the CDMO's development team, who works closely with teams like the MSAT and manufacturing team to make sure that as the process is developed, it's being developed in a way that is suitable for manufacturing. Also ensure you support a structured and milestone-driven tech transfer process. Sometimes the tech transfer can be endless if we don't put in the right milestones and the right success criteria. So definitely make sure that happens. And also trust the CMO's feedback and expertise. Remember what I said before, 2 brains are better than 1. So this is a very busy slide, but it's really showing you the tech transfer in a nutshell and what you would expect to see from, especially from a process perspective. So you'd see terms like trading runs and engineering runs and PPQ runs, aseptic process validation runs. There's a lot of structure that goes along a tech transfer when you're trying to bring something to the clinic. And that not only includes process, but it includes analytics and things like facility onboarding, raw material onboarding, and equipment onboarding. And these are all things that a good CDMO is really skilled at and can support you through the process to the clinic. If you want to read a little bit more and understand some of the other goals of successful cell therapy tech transfer and how you do that, we did publish an ebook a couple of years ago now, and so feel free to take a look at that. Definitely has some interesting points in there. And so really, I hope in this talk, I've explained how much a CDMO can help support a developer to get to the bedside and to do some of that clinical manufacturing. The goal of the CDMO too is to remove some of that burden for the development team. It's always a goal for the developers to be able to develop and for the manufacturers to be able to manufacture, because when you partner those two things together is really when I think you can succeed and bring products to the patients. And so with that, obviously working with the CDMO, how do you even begin to select the right one? And there's a few key points that I highly recommend. One is interviewing, right? Visit, research, ask all the questions. Don't just go on Google and say, hey, what CDMO should I work with? Actually get involved and speak to the people you'd be working with. Time and money are important, but shouldn't be the number one deciding factor. We're in an environment right now with excess capacity, so we are seeing some CDMOs that are underbidding just to get products in the door, but then aren't able to deliver because they don't have the right skill set. So just make sure that you're keeping that in mind when looking for a partner. Also, and this goes across the board, whether you're in development or tech transfer or working with the CDMO, but ensure robust communication and transparency. If you're having a problem with your process, let us know. Otherwise, we can't help. Definitely consider a CDMO that has technical expertise in your platform and cell type. Cell therapy manufacturing is still not your traditional manufacturing. It does require a technical understanding of cells and processes. So make sure you're interacting with a CDMO that has that skill set, and also with one that has an eye on innovation, both technically and operationally. So with that, I always ask the question, do you want your cell therapy program to be a big fish in a small pond or small fish in a big pond? And really what I mean by that is sometimes the smaller, lesser known, specialized CDMOs will pay attention to your program a lot more and have a lot more technical understanding than some of the large big box CDMOs that might be doing cell therapy, gene therapy, antibiotic manufacturing, antibody manufacturing. So definitely take a look at something that at CDMOs that are able to support your program in the way you need. And so with that too, I highlighted my career path, but I also want to highlight what that would look like for other scientists considering this path. It's definitely not a traditional path that's talked about in universities enough. So I do want to advocate for a path that provides a ton of knowledge and sometimes some accelerated expertise learning, because you are touching diverse science and technologies and get to have an end-to-end exposure with drug developers and things like compliance, regulatory, and the quality team. It allows you to have an impact at scale in a very mission-driven environment, right? We see patient material come in the door every day and out the door with product going back to patients. And I do want to say Roslin is hiring at both sites, so feel free to take a look at the openings that we have. And with that, I'm definitely open to anyone who wants to connect. So feel free to reach out to me either by email or through LinkedIn, and I will gladly have that conversation with you. And I hope you learned a little bit about what CDMOs can do to support your programs.
That was a fantastic talk. I mean, I particularly enjoyed the fact that you talked a little bit more about your career path and trajectory, I think that will resonate quite a bit with the attendees today. Go ahead and turn my camera on. If you do have any questions for Catherine, you can put them into the chat or Q&A box, and she can go ahead and answer them there. Otherwise, you can also email her. So I do have just a few kind of conclusion slides for everyone. So let me go ahead and share my screen one more time. Okay, so before we kind of sign off, I do want to just share some last few slides that highlight our humankind growth factors and how their potency, purity, and quality really do make them ideal for your discoveries and scaling up in cell therapy. So not only do we offer the largest portfolio of in-house manufactured GMP grade growth factors on the market, but we can seamlessly transition from research to GMP grade in order to facilitate a smooth transition from preclinical to clinical. Both our RUO and GMP products are manufactured using the same expression systems, production process, and operating procedures in a GMP facility. And so just to give you a little bit more information about our manufacturing, so we currently offer 80 RUO and 58 GMP grade cytokines and growth factors. And all our manufacturing is done at our headquarters in Illinois in order to avoid any sort of supply chain issues. We also have the infrastructure to provide our products from milligrams to gram levels. So as your therapy scales up, we can continue to provide you with the necessary materials. All our products are also manufactured using highly maintained equipment and in an ISO level 4 tested clean rooms. For our GMP products, we do adhere to rigorous quality process that is ISO 13485 certified and also compliant to US Pharmacopoeia and European Pharmacopoeia sections for ancillary materials used in cell and gene therapy. And then currently, just to give you a little bit more information about our customers, we do have 100 GMP customers with a large academic customer base. Many of our customers are in the CAR T, NK, and iPSC space and range from those who are, in the process of discovery all the way to phase 3 clinical trials. Our customers also value the quality of our products and have rated our proteins an overall 4.9 out of 5 stars. Throughout our CGMP manufacturing by our highly trained personnel, our process and products go through a series of quality checks and validations that are extensively documented, which can be observed here in the left column, which explains our GMP quality. And this documentation is available to our customers, and each GMP product comes with the following product-specific documents listed here on the right. I want to next highlight the advantages of our humankind proteins. So one of the key advantages is that our proteins are authentic human proteins because they are derived from human HEC293 cells. So they have native post-translational modifications and protein folding in order to optimize biological activity. This is often lost when proteins are expressed in other systems, for example, like E. coli. Because of their native structure, we have observed that humankind proteins have superior bioactivity and better stability, which means you'll get enhanced results and healthier cultures. Specifically, we do see advantages in cell expansion as well as differentiation protocols. And because of our quality in our manufacturing process, you will see that our proteins are animal component free, they're xeno-free, endotoxin free, as well as tag free. Our manufacturing process also offers superior lot-to-lot consistency, which can be observed here, where we tested 3 different lots of GMP FLT3L, and we tested their ability to stimulate dose-dependent proliferation in a human acute myeloid leukemia cell line. And you can see with the 3 lines that there's almost a complete overlay within these 3 lots, and their activity was nearly identical. And then I just want to touch a little bit more on, why the expression system matters and why we put so much emphasis on the fact that our proteins are derived from human cells. So here, what on the right, what this is showing is a comparison between our humankind TNF alpha compared to an E. coli derived TNF alpha. So for proteins that are expressed in non-human expression systems, some of the pain points are going to be things like the lack of authentic post-translational modifications, high endotoxin levels, improper protein folding, and lower bioactivity, which is what we observed with, our E. coli-derived TNF alpha. So with our E. coli-derived TNF alpha here, we see that it's actually expressed in monomers with no post-translational modifications. Whereas in our humankind system, we see that our TNF alpha is expressed in trimers, which is the most active form of TNF alpha. And we have proper post-translational modifications and proper protein folding that would essentially mimic what you would observe in human TNF alpha. So you can be confident that the protein that you're exposing your cells to is going to be equivalent to what you would observe in a human system. So here, next, I'm just going to give some examples of some proteins that we have available. So this example is showing that we have the most bioactive beta-cellulin on the market. So here we compared our beta-cellulin with two competitors and calculated dose-dependent proliferation of a fibroblast cell line. And you can see that our beta-cellulin was 50 times and 100 times more bioactive compared to our competitor products, again, emphasizing, the importance of the expression and the purification methods. And then I also want to highlight that we have the only authentic GMP grade BDNF on the market. You can see that it is 6 times more bioactive than our leading competitor and results in enhanced neuronal differentiation compared to E. coli derived BDNF. So you can see that these cells here have much more elaborate processes that essentially mimic what you would expect to see in neurons. And then finally, I also want to highlight our IL-2 protein, which has been optimized for the expansion of T cell subtypes. So in terms of bioactivity, we observed that it was 40 times more bioactive than our competitor's IL-2, which was bacterially derived. Also, here on the right, I'm showing some customer data that highlights that our IL-2 promotes greater levels of gamma delta T cell expansion and proliferation compared to the competitor IL-2. And then I'll just end with this list here that showcases our current GMP product offerings. So in particular, the ones that are highlighted in green signify GMP proteins where we are the only supplier on the market. We also have more GMP products in the works right now. So upcoming, we have our beta-cellulin and FGF20. So definitely stay tuned for those. So with that, I would like to thank everyone for joining today. Thank you so much to our speakers for their wonderful talks. It was great seeing everyone's, current work, and also Catherine, you sharing kind of more about RoslinCT and your career trajectory. And it really makes me happy to kind of see where the future lies for cell therapy. And it seems like we are all in very good hands. I do just want to mention that, as an attendee of today's workshop, you will be receiving 2 follow-up emails. One will have a recording of this webinar. You also can request a free sample of one of our humankind recombinant proteins. This is going to be from this selected list right here. And then you'll also receive a certificate of attendance. If you are eager to get your free sample now, you can also scan this QR code and claim it right away. But you will get those follow-up emails. And we do ask that you please allow 1 week for those follow-up emails to be sent. So with that, I would just like to thank everyone for attending. If you do have any questions for the speakers, feel free to email them. I've included their emails on this last slide here, or you can always email us at events@ptglab.com. So with that, I will go ahead and end the webinar. Again, just thank you everyone for attending. Thank you for your patience with all the technical difficulties. And I hope everyone has a great rest of your morning, afternoon, or evening, depending on where you're tuning in from the world. Take care, everyone.